Hello, my name is Corey Crawford, and today I will be presenting some preliminary research for the current project I'm working on, entitled Sex Differences in Morphine Withdrawal-Induced Anxiety, the Role of Estradiol and NMDA Receptors. Opioid use disorder, or OUD, affects millions of people worldwide. However, much of the research on withdrawal has focused primarily on males. We know that men and women experience addiction and withdrawal differently. Understanding these differences is key to improving treatment strategies. One factor that may play a significant role is estradiol, a hormone primarily found in females. Estradiol has been linked to emotional regulation and anxiety. So during opioid withdrawal, changes in estradiol levels may influence anxiety-like symptoms. With that being said, our main objectives were to compare the anxiety levels among males and females during opioid withdrawal, examine estradiol's role in withdrawal behaviors, and lastly, study the changes in NMDA expression in various key brain regions. We used a total of 29 adult long Evans rats, 17 males and 12 females, divided into two groups, one receiving morphine and the other receiving saline. Female rats underwent a daily lavage. The experimental groups received morphine sulfate injected subcutaneously twice daily for 10 days. The dosage was gradually increased from 2.5 milligrams per kilogram to 40 milligrams per kilogram. Control animals received injections of saline. To assess anxiety-like behavior, we use the elevated plus maze. We also analyze protein expression in key brain regions via Western blot. For anxiety, we examine the amygdala and ventral hippocampus. For addiction-related changes, we focus on the nucleus accumbens core and shell, as well as the ventral tegmental area. After 24 hours of morphine withdrawal, both sexes showed increased anxiety-like behavior. Notably, females exhibited a significantly greater increase in anxiety compared to males, suggesting a stronger withdrawal response. We then examined how estradiol levels influence anxiety during withdrawal. Interestingly, high estradiol levels were linked to increased anxiety during withdrawal. This suggests a shift in estradiol's role from anxiety reducing to anxiety increasing in the withdrawal state.
Hi, my name is Davis, and I'll be covering the Western blot results. We looked at the NMDA receptor changes in subunit expression following morphine withdrawal-induced anxiety. The key subunits are GLUN1, GLUN2A, and GLUN2B, which form part of the NMDA receptor. For the anxiety pathway, we found some differences between GLUN2A and GLUN2B subunit expression in the amygdala and ventral hippocampus, and for GLUN1, we found mainly in females that expression of the subunit increased after morphine withdrawal. GLUN2B had relevance in the nucleus accumbens shell with baseline differences between males and females, with females having greater levels that are reduced after morphine withdrawal. In conclusion, there were sex differences in anxiety after morphine withdrawal. Females were less anxious, and estradiol was found to increase anxiety after morphine withdrawal when separating females by stage of the estrous cycle. Females having greater levels of GLUN1 may be underlying their increased withdrawal experiences, and the differences in the subunit expression across the addiction and anxiety pathways show how the brain responds after withdrawal. Understanding differences between males and females and the role of estradiol will lead to a better understanding of treatment in women. Future research will investigate over-actimizing female rats and administering estradiol to investigate low and high estradiol effects on anxiety-like behavior after morphine withdrawal. Western blot will be done to see the protein expression changes, and another avenue of research is to investigate the role of other hormones such as progesterone in anxiety-like behavior.